So this video, we are discussing the bioeffects relating to time-varying gradient fields in MRI. The physical interactions that we're interested in are between the time-varying magnetic fields, or TVMF for short, and the biological tissue. And this interaction is described by Faraday's law of induction, where we have that the electric field produced due to a change time-varying magnetic field is equal to r over 2 times dB dt, where r is the radius of a circular loop of interest and dB dt is the rate of change of the magnetic field in Tesla per second. We can convert this to a current by multiplying by the tissue's electrical conductivity. Which is in Siemens. So we are going to look at several different aspects of this relationship. When we have a time-varying magnetic field due to the applied gradient, we can consider the waveform of the gradient. Typically, a gradient may take on the form of a trapezoid, meaning that it has a portion where it is rising and falling, and then a portion where it is steady. So this rising and falling is where the changing magnetic field effects are of concern. Whether the magnetic field is increasing or decreasing will result in different polarity fields. And ultimately, we are concerned about the electric field and current density that is produced in the patient during this time-varying magnetic field period. We can model the body as an elliptical cylinder with a semi-major axis A and a semi-minor axis B for the elliptical cross-section. If we do that, then we can calculate the maximum current density J to be equal to A squared B over A squared plus B squared times sigma, which is the electrical conductivity of the tissue, times dB dt. The electric field and the current induced in axial planes and their strengths depend on several factors, including the gradient strength and the slew rate, the position along the gradient axis, and then the size of the induction loop that is actually being considered. Current density J also depends on the electrical conductivity of the tissue. So these are electrical field factors, and this is another thing we need to think about when looking at the current. Ultimately, when we consider extremely low frequency, or ELF, time-varying magnetic fields, which are on the order of 10 to 3,000 hertz, we are most significantly concerned about the excitation of tissue, which may cause sensory or muscular effects in the body. The cellular effects have been examined through in vitro studies. Change in an outward flux of ions, specifically calcium ions, across the cell membrane have been observed. But the actual mechanisms for low frequency, low field effects are a bit difficult to pinpoint through research studies. But high field studies with human cells did not show any DNA damage or proliferation, which is something you might be concerned about if you're looking at cancer effects. There's also been a concern about 50 to 60 hertz electromagnetic fields and its association with cancer. A World Health Organization report recognized a weak correlation for magnetic field exposures above 0.4 microtesla at power transmission frequencies with the incidence of leukemia. However, this was a correlation observed and not necessarily a causation as far as the research went. And it's important to note that these these exposures were prolonged and chronic, and when compared to what you might get from an MRI examination, because those are more short term. Therapeutic magnetic stimulation, or TMS, has been used for bone healing since the 1980s. And although it's not entirely clear what its efficacy is, and the research regarding the mechanisms and effects are not completely conclusive, it is a method that has been used clinically for a while. Transcranial magnetic stimulation, which is actually what usually is referred to as TMS, arose from research relating to MRI effects relating to time-varying magnetic fields. And repetitive TMS has been used to treat psychiatric conditions to different degrees of success, but it has now at this point become an established technique to treat depression in some areas. So what happens with magnetic stimulation? Well, one effect is magnetophosphenes. So phosphene means to show light, and essentially magnetophosphenes are when you perceive light as a subject, and these like flashes of light, and the mechanism is thought to be through the stimulation of the retinal receptors or the optic nerve. The threshold for this is 12 millitesla RMS, or a peak-to-peak 34 millitesla. And the effects and the threshold do depend on the ambient conditions and dark adaptation of the subject. Magnetophosphene sensitive frequency range is not usually present in exposures, so this is not something that is frequently seen in MRI at all, but it is occasionally reported from movement near the high field systems rather than the imaging gradient time-varying magnetic field. It's important to consider, though, as they are considered by the International Commission on Non-Ionizing Radiation Protection, ICNIRP, to be the most sensitive acute sensory effect due to TVMF, so it is something that is considered when determining occupational exposure limits. Another magnetic stimulation effect that we have to consider is nerve and muscular stimulation. So the magnetophysiological relationship relating to this is described by what we call the strength duration curve, or SD curve. And there's several characteristics of this SD curve that are convenient to understand. So the minimum threshold, or Rio base, or RH for short, occurs for long stimulus durations, but for short stimulus durations are associated with an increased threshold. The chronaxy, or CH, stimulus is the stimulus duration for which the threshold is double the intensity of the Rio base. To understand the biological effects of time-varying magnetic fields, it might help to understand the excitable tissues that we are concerned about, including neurons. And so with neurons, we have a sequence of events that lead to the excitation. I would encourage you to look deeper into this if you have not studied it before. But the idea is we have a neuron, we have a cell body, we have an axon, and we have these segments of the axon that are insulated by the myelin sheath. And then in between those segments, there are gaps of insulation, which we call the nodes of Ranvier. And so the myelin sheath is there for insulation. The nodes in Ranvier is where excitation occurs, or the excitation is propagated from the different ends of the neuron to different neurons. And so the signal is propagated from these different points, nodes. The cell is at a resting potential of negative 70 millivolts, and this is maintained by two mechanisms, electrical forces and osmotic forces. Slowly the cell might be depolarized, but once it reached a certain threshold, sodium ion channels open and sodium ions enter the cell, further depolarizing